everyone, welcome back to our Medical Brain YouTube channel. So in this video we are going to discuss about the electrophoresis, which is one of the important topic. So the electrophoresis is nothing but the separation technique. So the movement of the charged particles in an electric field resulting in their migration towards the oppositely charged electrode is called as the electrophoresis. So in this technique, what happens is the positively charged ions will move towards the negative electrode and the negatively charged ions will move towards the positive electrode. So in this, there are three types of electrophoresis. So the first one is the zone electrophoresis. In that, there are two types, that is paper electrophoresis and the gel electrophoresis. Second one is isoelectric focusing. Third one is immunoelectrophoresis. So, zone electrophoresis, on what base or what the source we are using for the electrophoresis, on that basis there are two types. First one is the paper electrophoresis and the second one is the gel electrophoresis. So, in this, let us discuss about the paper electrophoresis. So, it is also a separation technique where we are used, it is used for the separation of the amino acids, proteins and the peptides. So, coming to the procedure, firstly, as the name it suggests, paper electrophoresis, we are using the Whatman number 30 filter paper. So, in this paper, at the center of this paper, we are going to load our sample, which consists of the amino acids, peptides or proteins. So, at this center of the paper, we are going to load the sample. After that, the edges or the tips of the paper are dipped into the buffer solution and further which are connected to the electrodes. Okay, that is, one is positive electrode and the here one is the negative electrode, and the term buffer refers to the solution which does not alter the pH of a solution. So after all this setup, what we have to do is we have to pass the electric current. So after some time, we have to remove the electric current and the paper will look like this. So here the first one or the center part is the place where we have loaded the sample, and after the effect of the electric field, what happens is the negative ions will move towards the positive electrode and the positive ions will move towards the negative electrode. So by this process, we are able to separate the negative ions as well as the positive ions. Then coming to the advantages of paper electrophoresis is it is a simple, easy and the cost effective procedure. And coming to the disadvantages, it takes a lot of time. Approximately it takes about 12 to 14 hours for this separation. And coming to the application, as I told you, it is used in the separation of amino acids, proteins and the peptides. And next we are going to discuss about the gel electrophoresis. Next type is gel electrophoresis. As name suggests, here the basic separation material used is a gel. Okay, so this is the setup of the gel electrophoresis. Here it is a apparatus where here the buffer solution is placed and it is a gel. Okay, commonly here the gel used is a agarose gel or the polyacrylamide gel. So what we have to do is we should place the gel material here, and in this the buffer solution is filled and here the electrodes are placed. So the electrodes are placed in such a way that the negative electrode is placed near the sample and the positive electrode should be placed far from the sample. Okay, so the principle is so simple, that is the larger molecules will move slowly and the smaller molecules will move faster towards the positive electrode, and hence separation will take place based on the molecular size as well as the charge of the sample fragments. So here in this one, here we are going to use the agarose gel which is in a rectangular shape. Firstly, what we have to do is we are going to dig some of the sample by using a comb like structured material. So we have to place or we have to put a comb here for certain time and the gel is solidified, and after that we have to remove that comb. Okay, so whatever the wells are formed here or well like structures are formed here, those are called as the sample wells. So in this sample wells we are going to load our sample material, okay, or the sample which we are going to test for. Okay, so here we are using agarose gel or the polyacrylamide gel. Okay, so after the electric field is applied, so here the sample fragments will move from the negative electrode towards the positive electrode. Okay, so the larger fragments will remain near to the sample well, whereas the smaller molecules will move faster and they will near to the positive electrode. So this is about the procedure of the gel electrophoresis. So in this technique we are going to separate the DNA, RNA and proteins also. So coming to the advantages of this gel electrophoresis is, it is a easy method, and here the gel or the agarose gel or the polyacrylamide gel used will give the accurate answer. And coming to the applications, usually it is used in the DNA fingerprinting and paternity testing, and the or it is in the application of the PCR technique, that is polymerase chain reaction, that is used in that is also a part of the DNA fingerprinting. So it is all about the electrophoresis, and its first type, that is zone electrophoresis, and in that we have discussed the two types, that is first one is paper electrophoresis and second one is the gel electrophoresis. So this section will end here, and thank you for watching this video, and please subscribe to our YouTube channel.